Alright, so lastly is chapter 8, and just to tell you what to expect out of chapter 8, it's all about DNA, RNA, and making proteins. And so this should be fairly simple. It's really going through your notes and answering the questions, and your worksheet should have also helped you with it. But let's just go ahead and take a look. Alright, so you all know that DNA contains your genes, or hopefully by now you do. DNA passes along genetic information, and it controls the synthesis of proteins. So we should all remember that DNA makes RNA, and RNA makes proteins. So it's a nucleic acid, remember, from our macromolecules. It has a sugar molecule called deoxyribose. RNA has a sugar molecule ribose. Remember, sugars N and OSE. It has a phosphate group, talking about DNA, and then it has the nitrogen in its base, adenine, guanine, cytosine, and thymine. It's double-stranded, and then it has hydrogen bonds between the bases, and the bases always pair up A to T and G to C. One of the most important things that you could ever in your life remember is that it always base pairs, and A pairs with T and G pairs with C. Like, if you do not know that, then you are quite frankly missing out on the whole thing. And again, knowing that hydrogen bonds are between the bases. And I somehow got stuck, so let me see if I can unstick myself. Alright, so DNA replication is a way that DNA makes more of itself. And so you should know that that's the way that we make more DNA, is by doing replication. Base pairing, of course, aids in replication. It says you have a DNA polymerase, which ends in ASE, so it's an enzyme, builds a new strand of DNA that will pair to the old strand. I'm so sorry. Alright, so this means that where there is an A on the old strand, polymerase will add a T, because A always pairs with T. And if there's a G on the old strand, then the new strand will have a C. So A pairs with T, G pairs with C. Something that you just have to remember. So there is something called the DNA code, the sequence of nucleotides that form the proteins. You have the 4 letters A, T, G, C that are used to make the words, but each word is written or read in 3-letter code. So the order of the amino acid is determined by the order of nucleotides in DNA. DNA gives us 3-letter codes that tells us what protein goes there. So when we talk about making protein, we have to talk about RNA. So we have DNA to make RNA, and then we have RNA to make proteins. RNA has, again, the sugar ribose. DNA has deoxyribose. And it contains the bases adenine, guanine, cytosine, and uracil. If you remember from previous lectures or from Science 100, DNA has thymine, RNA has uracil. So in DNA, A pairs with T, in RNA, A pairs with U. So there are 3 types of RNA. Remember, looking at the 3 bullets at the bottom, there's mRNA, tRNA, and rRNA. And so remembering those, that rRNA is what ribosomes are made of. I'm just going to go back. tRNA transfers the amino acids to make the proteins. And then messenger RNA is your main RNA used to tell what protein is being made. So transcription, of course, is the process of going from DNA to RNA. So you should remember that. And then this just talks about the process of transcription. I don't go through that as much. Translation is taking RNA in making proteins. And as we said, there are 3 types of RNA, but messenger RNA is the only one that is read so that proteins can be made. And this is a chart of the genetic code showing you when you have 3 letters, what amino acid goes with those 3 letters. And as I said, RNA is found in ribosomes. And we talked about before how the ribosomes are where, I'm sorry, I'm very tired. That ribosomes is where your messenger RNA and tRNA meet. And this is how a beautiful protein is made. And so I don't go through all this, but you should know that in order to initiate translation, you need this 3rd bullet, the start codon AUG. And AUG tells proteins to start being made. So it's known as the start codon. And so once you start, you then elongate by adding amino acids onto it. And then to end it, you have these 3 stop codons, UAA, UAG, and UGA. So you should know for translation that there is always a stop and a start codon. And then you terminate, and that is it. As I said before, you need, for each amino acid, there are 3 letters like AUC, AUG, TCC. Those are 3 letters. So if I have 12 of those letters, that means that I can make 4 amino acids because every 3 letters gives me 1 amino acid. And I'm pointing that out because you'll have questions that ask you things like that. If you have so many letters, sequences, then how many amino acids you can make. And you just have to remember that each amino acid is made out of 3 letters. All right. And so I don't think we talked about protein quantity and packing. But I would go over these just in case there is a question or 2 that comes up. You may have questions about mutations. And there are 3 different types. I would go over those just to make sure. Knowing that sickle cell anemia is a point mutation. And then you have your frame shifts. And I do not remember doing chromosomal aberrations. All right, so that ends chapter 8. And I think I have helped you out plenty with that one. All right.